Okay. So, hello, my name is Rana Nasser and my colleagues Salim Siblani, Jana Shukr and Ali Fawaz will be presenting about the oxidative phosphorylation dependent regulation of cancer cell apoptosis in response to anti-cancer agents. Now, the main idea of this article is to point out the mitochondrial targeting in cancer cell therapy depending on the oxidative phosphorylation pathway, which will be annotated by OXPHOS. And knowing that the mitochondria regulates signaling pathways in cancer cells, researchers used this fact in the therapeutic field where anti-cancer agents with different classes were used to induce apoptosis by affecting these pathways. And now I will provide you with an overview about OXPHOS. OXPHOS is an active form of respiration and is made up of different complexes encoded by mitochondrial DNA and nuclear DNA, where mitochondrial DNA specifically showed great importance in apoptosis, and any mutation in the mitochondrial DNA will lead to OXPHOS impairing. Now, moving on with the main methods and materials that were used, RNA sequence analysis, Western blot, analysis of mitochondrial DNA by real-time PCR, flow cytometry, and statistical analysis by INNOVA. Coming to the results now, diverse anti-cancer agents show cell type dependent effects, and by this I mean that they use two types of cancer cells, prostate cancer cells and colon cancer cells, both treated with anti-cancer agents. These agents in the prostate cancer cells induced non-apoptotic cell death necrosis, while in the colon cancer cells they induced caspase-dependent pathway to induce apoptosis. Not only this, but also for the same cancer cell type, these agents exert differential sensitivities. As for HCT 116 cells, apicidine induced the highest percentage of cell death, 28%, and tepsigargen scored the least percentage of cell death, which was 12%. And now for etoposide. As for etoposide DNA damaging agent, it induces an increase in the mitochondrial DNA, and this can be clearly seen through the increase in the expression of important genes, just like ATPase8 and COX2, that are important for apoptosis. But also it is good to mention that OXPHOS complexes, specifically complex 1, 3, and 5, they play a key role in apoptosis. Because upon the addition of the inhibitors of these complexes, a significant downregulation in the mitochondrial DNA content was seen. Now for the P53, which is a tumor suppressor gene, it has a combinatorial effect with etoposide to increase the mitochondrial DNA content. Moving on to ROS. ROS is the reactive oxygen species that play a role in cellular oxidation metabolism affects cell death as well as cell differentiation. The elevated ROS production induced by etoposide triggers also an increase in the mitochondrial biogenesis and mitochondrial mass, thus favoring apoptosis. And this can be clearly seen here in the three graphs where we have a comparison between the control cells and cells treated with etoposide. And now we'll move on with Salim.
Hello, I'm Salim, and I'll be presenting the experiments that were used for the investigations of these agents. In this experiment, we tested the roles of autophagy and caspase on mitochondrial biogenesis. We applied experiments using one inhibitor for autophagy and one inhibitor for caspase. The results of Western blot to the right side of the screen showed lower levels of POLQ and TWINKLE upon the inhibition of autophagy activity, but not the caspase activity, noting that POLQ and TWINKLE are mitochondrial DNA polymerases. So inhibition of autophagy, but not caspase, downregulates mitochondrial biogenesis. This experiment tests the effect of etoposide on heat shock proteins that are important for mitochondrial UPR. The results show that HSP levels were elevated upon the treatment with etoposide as shown in the diagram. So etoposide upregulates HSP that are essential for mitochondrial UPR. This experiment shows the transcription faults of mitochondrial content upon treatment with either doxorubicin or etoposide, in which doxorubicin downregulated all OXPHOS transcripts except 7 of them, while etoposide downregulated all of them, and that's the major difference between both agents on OXPHOS metabolism. For this experiment, we tested the role of complex 1 in caspase-dependent apoptosis. So we tested the effect of etoposide on all cells lacking complex 1, which showed lower levels of caspase activity. So simply we can say that complex 1 plays an important role in caspase-dependent apoptosis. In this experiment, we tested the role of both complex 1 and OXPHOS in 4 different agents. So we used cells that lack either complex 1 or OXPHOS fragments. The results show that etoposide, apicidine, thapsigargen induce caspase activation, while Taxol and sorafenib have caspase-independent pathways. Complex 1 deficiency and the gross OXPHOS deficiency reduced caspase level in response to thapsigargen, while it was increased in response to apicidine. And now, Jana, the stage is yours.
Hi, I'm Jana, and this slide I'll be explaining 3 experiments. Now, in the first experiment with the 3 graphs, we wanted to determine the percentage of apoptosis in the wild-type cells and complex 1 deficient cells with different anti-cancer agents. Now, in each of the graphs, with doxorubin, tapisartan, and etoposide, respectively, the percentage of apoptosis was reduced in complex 1 deficient cells, G18 and B2, in comparison to the wild-type cells, G3 and B1, which shows that complex 1 shows a great importance and role in inducing apoptosis in response to anti-cancer agents. Now, moving forward to the second experiment, showing the result of a Western blot that was applied to determine the level of release of cytochrome C from cytosol with doxorubin presence. We must mention that the cytochrome C is released due to the destabilization of mitochondrial membrane by ROS increase. This release leads to caspase activity and apoptosis enhancement, especially when doxorubicin was present with the wild-type cells. Moreover, looking to the second Western blot, here the cytosol and mitochondria were isolated to measure the release of AIF, which is the apoptosis-inducing factor, which was downregulated with complex 1 deficient cells, G18, with respect to the anti-cancer agents in comparison to G3 and G7 cells. So from the 2 Western blots, we can say that complex 1 deficiency affects the release of AIF and cytochrome C, thus inhibiting apoptosis. Now, in this experiment, we measured the caspase activity with respect to doxorubin and rotentin presence. We noticed that when rotentin was present alone, as rotentin amount is induced, caspase activity increased. But when doxorubin was present with rotentin, we noticed that when rotentin amount was elevated, caspase activity was reduced. So we can say that rotentin alone interfere with the electron transfer chain by inhibiting it, generating ROS increase and mitochondrial biogenesis, therefore activating caspase activity and apoptosis. In this figure, we wanted to determine the doxorubin affinity of binding with respect to the wild-type complex 1 deficiency and complex 2 deficient cells. We noticed from the first graph with the wild-type cells, and also with respect to the table, that there was binding present. Now, looking at the second graph, with the complex 1 deficient cells, no binding was detected. And there was binding detected in the third graph with the complex 2 deficient cells. So we can say that DNA damaging agent doxorubin binds to the molecular targets in complex 1 to initiate apoptosis. Now, Ali.
So in reference to the previous experiments conducting anti-cancer agents and their way of acting to induce apoptosis in cancerous cells, we can say that these agents work either by a mitochondrial DNA-dependent pathway or a mitochondrial DNA-independent pathway. Moreover, combinational therapies are of a great importance in this manner because they can increase the induction of apoptosis in these cells by using combination of several anti-cancer agents. But a great concern about these combinational therapies is the high level of toxicity due to the usage of several anti-cancer agents at the same time. So a pathway of metamorphin in parallel with other non-DNA damaging agent is a choice which can decrease the level of toxicity of these combinational therapies. Now, talking about the anti-cancer agents that we've studied in this research, we should say first clearly that there's some of them that do not have a very clear way or mechanism so that they can induce apoptosis through oxidative phosphorylation dependent manner through mitochondrial biogenesis, including apicidin and sorafenib. On the other hand, we studied the way of etoposide induction and doxorubicin to induce apoptosis in cancerous cells. So we can say clearly from the given experiments conducted that etoposide acts like a DNA damaging agent, which damages DNA that leads to the expression of a tumor suppressor gene, P53, that downregulates AMPK that activates the mitochondrial biogenesis and increases mitochondrial DNA through autophagy-dependent pathway, which leads to the increase of reactive oxygen species in the cytoplasm secreted by the mitochondria, which leads to apoptosis. And another novel mechanism held by doxorubicin, which is characterized by the binding of doxorubicin to complex 1, which is a mitochondrial membrane protein, leads to a conformational change in this complex, which leads to the high secretion of reactive oxygen species and which leads to apoptosis. And it's worth to mention here that all of these agents induce apoptosis after mitochondrial biogenesis and elevation of ROS through caspase-dependent manner and cytochrome C. So to sum up all of the information, we can conclude from this research that in the future we should have more specific combinational therapies using specific anti-cancer agents to hold in parallel and a good way to induce apoptosis in cancerous cells. That's all for the presentation. Thanks for your time.
Thank you.